Hi, my name is Christina Simkanin, and I'm a graduate student working on my doctorate at the University of Victoria in British Columbia, Canada. And I first met Jennifer Dijkstra, my project partner and collaborator on this project, in June 2011 at the NSF-funded PASI Advanced Tunicate Biology and Taxonomy course that was carried out at the Smithsonian Tropical Research Institute in Bocas, Toro, Panama. And as part of the course, Jen and I worked on a student project that we then carried on looking at by traveling back to Bocas del Toro in November 2011. And our project is looking at how osmotic stress or fluctuations in salinity affect the physiology and growth of two tropical botryllid ascidians. And the two ascidians that we studied were Botryllus planus and Botryllid nigrum. And the three metrics we used to measure stress due to salinity fluctuations were heart rates. So we looked at how heart rate was affected. We also looked at how the rate of asexual reproduction or zooid growth was affected by salinity. And then we also looked at overall growth of the colony. To get started, before we could actually carry out our trials, we traveled to the Bocas del Toro marina to collect our specimens, and luckily both of these botryllid ascidians are fairly abundant there, and they grow on both the dock floats and the pilings. So we traveled to the marina and removed the ascidians using scrapers and other implements. We brought the ascidians alive in coolers back to the marine station, where we had the colonies resettle and attach themselves onto glass slides. And the utility of glass slides is that you can easily move them around from one sort of experimental trial to another, and also to examine internal physiology characteristics in colonial ascidians like heart rates or asexual budding. The glass slides allow you to sort of have a window into the actual colony itself. So if you flip the glass slide over onto a microscope, using a dissecting microscope, you can count heart rates. So after collecting the ascidians and having them settle onto the glass slides, we set up our experiments. And to test the effects of salinity, we used 5 different salinity levels. So we used 20 PSUs, 24, 28, 32, and 35. And we had 5 replicates at each salinity for each species. And our experiments were run for 10 days outside at the aquaria at Stry, and so they were at ambient temperature. And every day we measured salinity and kept salinity constant, so we would change the water if needed. We fed the ascidians using an aquatic invertebrate feed, and we took the ascidians into the lab and had a look at them under the microscope. And under the microscope we did timed counts of heart rates, and we judged what stage of their asexual reproduction cycle they were at. And we also took digital photographs so that we can look at growth during the 10-day experimental trial. And our preliminary results suggest that there's definitely differences between the tolerances of the two species. So Botryllid nigrum is less able to withstand the 20 PSU, so our very low salinity treatment, whereas Botryllus planus seemed more adapted and more able to survive at these lower salinities. And we also found that Botryllus planus seemed to sort of adapt this way of kind of closing itself off from the environment. So some of the zooids created coverings over the cloacal cavity, which we think basically allows them to regulate osmotic pressure inside the colony while keeping low salinity water outside. So they were able to close themselves off and still carry out their metabolic functions within the zoo itself, but basically stop feeding for a period of time by closing themselves off from the outside environment.
My name is Dijkstra, and I'm a postdoctoral research fellow at the Wells National Estuarine Research Reserve in Maine. And so the overarching goal of our research project is to really look at how low salinity events would affect population growth and distribution of these colonial ascidians. And the reason we focused on colonial ascidians is because, one, in other parts of the world, they're a nuisance or a pest species that people just want to get rid of. Two, they're pretty easy for us to culture. And three, they overgrow other invertebrate species in subtidal communities. And so for that reason, we're very interested in knowing the physical parameters that will either restrict their distribution, will limit their population, and even this may even help us to figure out ways to eradicate them.